Hello, everybody. Welcome to the CHI Virtual Speaker Series. Today we have Dr. Edwin Wang with us here. He is an undergraduate training in computer science and a PhD training in molecular genetics from UBC. After a one-year postdoc training at FlyBase, a database of Drosophila genes and genomes, he moved to the National Research Council as a PI. In 2016, he became an AISH Translational Chair Professor in Cancer Genetics at the University of Calgary. And today, he is a professor in the departments of oncology, medical genetics, and biochemistry and molecular biology. In his lab, he and his team are conducting both computational and experimental systems biology towards precision medicine. The computational, sorry.
I can stop. Yeah, can stop shared.
Sorry about that. It just covered my screen. His work encompasses big medical data analysis, machine learning and deep learning, and predictive model construction based on genomic data of disease, including cancer. And from there, we can screen share and start the presentation with Dr. Wang.
Okay, so first, thanks for the nice introduction. Also, thanks Hood invited me, gave a talk here. Just try to share some of my work. Probably I can give you some idea what I'm doing here. So, as explained, I basically did lots of computational work before moving to Calgary. And now, starting from 4 years ago, I started 2 labs in Calgary, University of Calgary. So one is still maintains our roughly 80% of activity in computation, but 20% activity in really wet labs. But today I will not talk about anything for wet labs. I'm really talking about the computational part. So computation part, we are more interested in using machine learning models and deep learning models to do translational medicine studies. So here, so for the people maybe don't know some people always ask me, so where your wet labs? Yeah, my wet labs, I think near the building of you guys. And the current activity majorly working on the single cell genomics. So this is really pretty new technology. It's a few years. I think so far in the university only maybe 5, 6 labs they working on single cell experiments, and we are the only labs working on the secondary generation of single cell studies. This is can be 100 times cheaper than the first generation. So that's yeah. So one of the I mentioned this because later on I will need this stuff. So one thing is that we can use the ATAC-seq data. This is one of the technology, sequencing technology, allowing us to build up the gene regulation network. As long as we have the data, we can really build up a network, a data-driven networks. So that's just wet lab working. So I stop here for wet lab part. I just gave you some rough idea. So what are we doing their part? And let's back to the computation. So the computation, one of the observation, as many people realize that omics revolution, that means large-scale, high-throughput technology in medicine and biology, and really generate lots of data. And they will continuously to do more. And based on those data, you can start with personalized medicine. This is, 5 years ago, at that time, President Obama launched this. And also at that time, we thought about, we wrote a paper, a sort of perspective, to talk about, we predicted that at that time, maybe 5 or 6 years later, the sequencing cost should be pretty low. And what happened? What we should do? If we have lots of those data. So we propose some conceptual framework, how to build up models and to using those data. So that's, you can, from the title, you can see that. And it's not surprisingly, but interestingly, you can see that in 2016, if you sequence one human genome, costs $1,000 US. But today, $100. So $100, this is technology already in Illumina, but they didn't put it on the market yet because they want to sell their current sequencers. But on the market, you can get $200 at the moment for one human genome sequencing. So going on this, you can realize this thing can be used in the clinics or even in the population. $100 for sequence whole genome, you know, so what? Really cheap. And even I can expect it maybe next 5 years. Within the next 5 years, that can be shrunk down $20 to $50 for one human genome. So in that case, that's much cheaper even do a test, just a simple test, even X-ray test, for example, in the hospital. So you can see a large amount of such data will be generated easily and scaled a lot in the future. And then the question is coming that the challenge is that how we can use those data to build up pretty models, right? This is challenging because there's so lots of data and you need to link to the phenotype of diseases, for example. So at that time, like I mentioned back to this paper in 2015, we proposed some protocol or framework to do this, right? I just gave you a few examples. For example, we can using that framework to modeling the networks and then predict drug targets. And then we can using that concept to build up the predictive models. So it means that you can predict who will, this patient, cancer patient, who can get, because once you get a cancer patient there, you don't know 5 years or 10 years, this patient will get metastasis or not. So you can do the prediction at that time before they get really metastasis. So the treatment can be different. So one of the real example here, I gave you one example. So for example, the colon cancer. So colon cancer basically, they have four different stages. So cancer in general have four stages, 1, 2, 3, 4. So the stage 4 normally is metastasis to many different organs. It's really hard to treat. So stage 3 have local metastasis, but not distant metastasis yet. Stage 2 have no metastasis yet. So traditionally, they have a chemo for colon cancer. They're really effective in stage 3. Means that when the people receive that treatment, roughly they have 30% of people, the patient, will get 5 years longer than untreated. But they try to use this in stage 2 patient. So stage 2 patient basically have 25% of the total colon cancer patient. And they did many clinical trials and found no differences. So treated and non-treated with the same drug, there are no differences. And then, so they have guidelines because cancer treatment often have guidelines. So European guidelines suggest stage 2 colon cancer patient, you don't need to treat because no clinical benefits. US and Canada guidelines suggest you treated high risk patients. However, there are no any indication who will be the high risk patient. So that's the problem. So we're starting this problem. So the question is that can we, using those technology, build up machine learning models to pick up the fraction of the stage 2 colon cancer patient. They are high risk to get metastasis, and then so that they can get a treatment, right? So that's the one is a example. So we are doing, and we build up such model. We can stratify a fragment of the stage 2 colon cancer patient, and then we compare their treatment. So this is, if you put all the stage 2 colon cancer patient together, the drug treatment control and treated people, there are no differences. However, you really see the big differences once you have those, once you shrink down this high risk group based on our sort of biomarker developed based on the machine learning. And then if you define, if treated and untreated, they have big differences. So they have 5 years survival differences. It means that you really can pick up a set of so-called high-risk patients from stage 2 and then really treat them. And other part is low-risk part. You don't need to treat because we saw that here with the low-risk people, when you put there, you treated and non-treated, there are not much differences with the drug. So this is one example. You really can, using machine learning to build up models and really apply it to the clinical setting, right? So, so another thing is that we go back to the sequencing data. So when we go back to sequencing data, when we think about, if you go to the, you go to the basic biology, you will find information flow basically from genotype, right? So that's the sequencing. And go to the molecular phenotype like gene expressions, and the cellular phenotype. So that is different cell shapes, and clinical phenotype is a different disease. So you can see this information flow. But when we really think about that, this is coded in black color. We find they don't have another phenotype, should call the gene regulatory phenotype. This is the red one we proposed. So we think this one should be between the genotype and the molecular phenotype. So how to model this? So how do we can infer such information of the gene regulation phenotype, right? So that's our concept. So we propose some network method to really infer such and the phenotype. And then we can model them based on a called a system biology concept that we call the tipping point. Is that suddenly a state to another state changing? That's the model points, right? So I gave one example. So we developed a algorithm called e-tumor metastasis. So this one is basically using the blood sample sequencing data. So let's say this is a patient, cancer patient. You take the blood sample. So this is a normal sample. This is not a tumor sample. So this is a normal sample. You sequence them, and we want to develop model using those sequencing data of the patient from blood sample, and to predict whether this patient will have metastasis or not. So that's data we use TCGA. This is a US cohort for near 10,000 sequencing of cancer patient of different cancer types. So we use those data. So we built up a new technology that we built up the first gene regulation network or signaling network for that cancer, let's say breast cancer. So that's based on the information, we can build such networks. The idea is that, so for the mutation, they are, for each patient, they're not same. So for example, for patient 1, this just give one example. They have many other mutations, but I just give you one example to show the concept. So for example, the patient 1, they have this gene get mutated. So this network is that each node is a gene, is a link, is between the gene relationship, okay, or regulation relationship. So the red one here is mutated genes. Okay, so this one have one gene get mutated, but another patient get another gene get mutated. So here, the network is same. So the gene changing, the mutation gene are changing. So that's exactly in the cancer mutation. So their mutation is always changing from one patient to another patient. So based on the machine learning, that will be really hard because machine learning is much looking for certain common features between one group and another common feature in another group, but these two groups, they have differences in these features. Because those mutations in cancer is so diverse, so you never find so that's challenging. So you never find a mutation really across all this patient, even same cancer, let's say breast cancer. You cannot find it, right? So that's really the challenging using the mutation data to do the machine learning prediction. So our strategy is that using this network, you can catch, for example, if you merge these three patient mutation, so you can really find that those mutation genes, they form a cluster. They're together. Okay, so that is to have an underlying mechanism is that because, for example, if one pathway, let's say you have 10 genes in this pathway. You can trigger mutation, any of them, you can trigger this pathway on. So it means that, so that the pathway can be on, activated, but the mutation genes can be differently from patient to patient. So that is a linear pathway, but in the, we model means in the network. So the pretty similar concept is that you can find it, you put them together, you can see this. So then we have using a, they call it network of propagation. This is the algorithm. Allow me, let's say, for example, starting from this point. So at the very beginning, so this is 1. I put this is 1, for example, gave a number 1. All other nodes give 0. Okay, this is starting point. And then what we can do is that along those energy, so let's assume this is the energy. So all 0 here at the very beginning. Here is 1. So because this is really high energy, so the energy needs to be diffused to other places. Okay, but they cannot diffuse what they want. They must be follow the route, follow those. And then finally, you will find that they stabilized because the energy diffused to some time point. They stabilized. When they stabilized, so each other node, they will get a score. Or we can get different scores. And then you can see that in this case, if you do that, you can find those nodes, they get higher scores in general. And this one get high scores. And this one get high scores. So this one is resemble those, pretty similar. So based on this, you can see, because those we can transform, let's say, that's each node is 1 gene, that's the row here. And each patient, this is a column. And the number here is that the score I'm talking about. So here, the same setting, but here the mutated ones always get 1, right? Another 0. But after you do this algorithm, you will find those genes, they get pretty similar scores. They're pretty similar. So that those patients, they have those kind of mutations. I can sort of make them similar based on these scores. So now you can find common features, right? Otherwise, you cannot do it. So that's a network-based idea and the concept. So here is just the example. So for example, we have the training set. Recurrent sample means they are metastasis sample. Non-metastasis sample, we use this network, we call it network profiling. Finally, you get those profiles, just like the matrix, right? Based on these 2 matrix, we can use our MS, this is before we developed the algorithm to get those signatures. So this signature really can do the prediction. So we can do the validation. So this is really whole exome sequencing data from blood sample only. And then we can do validate it. They can predict very well. So let's give you one example is that we have using this sequencing data to based on the genes, because of these mutations based on the genes. Those are all the mutations in genes. We build up models. So next one, I just gave you another example as the concept level is much more advanced than what I talk about. So far what I talk about, I call it, this is a traditional machine learning concept in medicine or biology. Now I'm talking about a new concept. It's called, we give a name. This name is, you can maybe you cannot find in the public place yet. They call it digital genomics. So I will explain what this means. So back to the diseases or biological trait, for example. We know height, for example. This is a trait, right? This is a biological trait. And before we thought maybe there's complex. They could have maybe 100 genes control this trait, right? Or 200, a few hundred genes control this trait. But when really people did study showing that, for example, you have 10,000, more than 10,000 SNPs, means you have in the genome, you have more than 10,000 genetic places, really can control the height, this kind of one of the biological traits, right? So it's really complex. It's not like we saw it before, right? So if based on the current study, if you try to understand how these 10,000 genetic places contribute this one biological trait, they maybe take us 200 years to understand this. This is just one trait. If we think about so many diseases are so complex, and other trait, for example, IQ, a disease of cancer. So there are a lot of those kind of complex trait or diseases. There will be a lot of such genetic places to control that. They may be, if using the current biological studies, the method, the ways to do, maybe take us another 1,000, 2,000 years, try to understand how they treat this, right? So we cannot wait that long, right? That's too long. So how we can do that without really understanding them yet, okay? So that's give one concept we call DNA fingerprint. So what does it mean, the DNA fingerprint? I'll give one example. If you have take an article, you read it, and they have ways to generate, they call it word cloud. Okay, word cloud is the frequency of the word representative to this article, right? So if you get, let's say, 100 articles, and all of them, they talk about World War II, for example. This is the topic of the article. But it's written by different 100 authors. However, when you generate the word cloud from each of those paper articles, you will get pretty similar pattern, or you can get a similar word cloud. So it means that this can summarize the story. Doesn't matter the story, it must be exactly the same, be written or be written by different authors. Okay, so if you see in that way, you really can think the genome as a book. So each individual can have a genome. So each genome is a book, right? So each genome contains, let's say, 10,000 chapters. Each genome contains, so each book have 10,000 chapters. So each chapter talking about one story. But from person to person, those stories could be changed. right? That can be changed, but some story pretty similar, right? Okay, just give one example. So if I take 10,000 books, means I take 10,000 genomes, 10,000 individuals' genomes. So each person have, let's say, 1,000 chapters, right? However, all these 10,000 people, they have one chapter talking about cancer, right? Everybody have cancer. So this 10,000 people I pick up is that everybody already have cancer. So it means that at least all of them have one chapter in their genome talking about the cancer. Okay. So if we can develop some method to extract the word code for that chapter, and then we can get so-called DNA fingerprint. So it means that those word code, or we can call DNA fingerprint, can link to cancer. Also can link to cancer. So once we can do that, we can do the prediction later on using this as a feature, right? This is the concept. So then I just gave you one, just recently published paper. We just published a paper, just apply this concept to do this. Well, that's also did this 3 years ago. Actually the work done 3 years ago, but just published 2 months ago, or 1 month ago, maybe 1 month ago officially. So what we did is that we take exactly 10,000 patients. So this is sequenced in TCGA. So, but we take their sequence or normal sample sequences, not a tumor, but normal. So normal sequencing, that means this represents the germline genomes of the patient. And of course, we have another controls. This is from another data set. This is another more, people didn't get cancer. So we apply a machine learning technology called NMF to extract the DNA fingerprint. So what the DNA fingerprint here looks like is that, so we starting look at the triplets, for example, in the genome, like a sleeping window, and then we ask question. Let's say in the middle one, they change to A, for example, from C to A, and what's the frequency in the genome, right? And then you put them together to see whether this pattern appear again in the chapter of the cancer story. So that's 10,000 patient genomes. So the idea is that, so finally I will say we found 7 different fingerprints, and these fingerprints really can represent it for each patient. You can get the fraction of the contribution for their genomes. So this is the idea of that. You pull all them together. This is all these the triplets in the 10,000 people. So that's the 10,000 cancer patient. So for example, here is the 7 fingerprint from the cancer patient. So we thought this is associated with cancer, whatever, right? This is, we call it cancer associated fingerprint. So one example, for example, we saw we saw that. So let's see if this is different cancers. So if this is the different cancers, right? And then they have there's two figures here, and this is one of the fingerprint. We call it E, fingerprint E. So fingerprint E, they appear differently in people they have cancer and they don't have cancer. So this always the first one, they don't have cancer is control, and the different color ones, these are different cancers. You can see the difference of the E in the genome. This is the contribution of E in the genome. That's the fraction of E in the genome. And then you see if people have this one, if they smoke, they tend to get cancer. So if they don't have this one, even they smoke, they don't get cancer. So this means that you really find a pattern there. So this pattern represents to interact with smoking to do that. So that's one example how that works. This is one example of that. And also you can see if I use the cluster analysis, because this is the seven, this is the 7 fingerprint. So each of the color here, so this is the column is samples, the column samples. So the color here is represents the fraction of each of this fingerprint in the genome, the fraction of that. So after normalize them, you do the cluster analysis. You really can define the patient. This is a brain tumor. So it's a brain tumor to 3 groups. And then you can find they have their survival is difference. Okay. So this is showing that you can use those data, you can do the prediction. So you can do, this is the survival, you can see difference from the groups, for example, here. Other cancers also, I didn't show that, we didn't show it in this talk, but other cancer, we also have shown this. But here the idea is showing that you really can extract certain DNA fingerprint, we call digital genomes approach to get this point. So that is sort of, we push the concept a step further from the current only focus on genes, because currently all the study, all this machine learning study is focused on genes or focused on the SNPs of that. But really they don't go to the, we talk about the fingerprint level. So this is one example. We try to push them out. So yeah, like I mentioned, so digital genomes, they talk about the study of the genome, the patients, they can link to the annotated genes. So even you see in the DNA, let's say promoter region, there's a regulation for genes, but still gene-centered is also the gene concept related. So digital genome seeking to link genome patterns or DNA fingerprints, the forgettable genes, to link certain traits and phenotypes. So that's different from the I mean, in the past, the concept. So we have currently working, so that's, like I said, this is 3 years ago, we starting from the digital genomes efforts. But now we are looking for digital genome 2, version 2. So this version 2 will combine the single cell ATAC data. If you remember, I mentioned my lab working on the ATAC data. And current trends, we predict that in 5 years later, so you will find a lot of such data for single cell, each single cell in even the different development stages, right? From embryonic stages, the cells in that stage, in fetal stages, the children and organs. So you will find out in the public set or in many labs, cannot be generated from our lab. There could be many labs in the world. They will generate such single cell data for each different stages for many cell types. But now we don't wait 5 years later, we do something. Now we should think about what we can do with this data set. Just like 5 years ago, we talked about if the human genome sequencing have there, what we should do. So then we developed this and this and this. So now we're thinking about another 5 years. So another 5 years, if such single cell level data generated, what we can do for that, how we can do it, right? So now we're using this digital genome concept, but the good news here is that the single cell ATAC data and a single cell RNA-seq data, you can use those to build up molecular networks or gene regulatory networks for each cells, for each individual cells. So we are working on a new algorithm, basically try to integrate the genome sequencing data with such molecular networks in different cells to model the diseases and the biological trace. So that's the current in my lab focus. So we are doing such stuff. And also, next we will talk about from digital genomes to digital health, we will integrate clinical information or EMRs, okay? So just give one example, we doing some work in the not genome set, just the clinical data set, okay? So the one is called deep learning, right? So deep learning, this back to a few years, they started, right? So one of the idea of deep learning totally are really different from the machine learning in many cases, is that they try to model the distance with nonlinear. So here, one example, this is slides from my students. So that's, you can see it's different background from other slides. So you can see their distance between the two places can be nonlinear. It's not modeled by linear. So linear, you can see like this, right? That's a linear. But basically nonlinear is quite longer than this one. For example, just give one example how this deep learning different from machine learning. And then we use one of the algorithm called autoencoder. So the idea that you can really encode the features, and then the output will be decoded. And then this is the hidden layer. So this is, you can have normally 3 or 5 layers, can be many layers based on the situation. So one example I'm showing is that here we use we don't have this one don't have genome data yet. So our ultimate goal is we will use the EMRs with the genome together. Here's only use the because from UK Biobank Data, it's half of them. This population for training. So we use the EMR features only here. We only use the EMR, no genomes yet. So only EMR data. We do the prediction of the cancer risk because they have cancer population here also. We can do this and validate it. So far it's not too bad, but we think when we combine with genomic data later on, so there will get much better model because they have interactions between the lifestyles, for example, with the genomics. So this one have no genomics part, but this one only use the deep learning technology. So the last few slides I just show you, we try to move into from digital genomes to digital health. So Alberta, we know they have richest data province in Canada. They have lots of electronic macro data. Like I said, in the future, genome sequencing will be easily implemented. So you can see in the future, maybe a few years, if you can easily combine genomic data and EMR together in the cohorts, for example, in Alberta. So now what we doing is that we use UK Biobank Data. So UK Biobank Data, if for somebody maybe don't know, is that this is a longitudinal study, and everybody, their disease is encoded there, even their other clinical information and lifestyle information also is there. And in Canada, we have another one called Campus. So this is another cohort. The UK Biobank Data, they have genome sequencing ongoing. So far, we have, I think this year, we get the data for 200K UK participants in the UK Biobank sequenced. And next year, all of them will be sequenced. Now, I'm doing sort of work, try to bring some dollars from government, from industry, working with Campus, try to sequencing the Canadian cohort in the future. We try to get that. So that this is the idea. So we can using this cohort to do the studies. And later on, I think they can apply to the really the real population in Alberta or in other provinces in the world, for example. Basically, you can combine the EMR data and the genomic data to build such models, and then to predict diseases, whatever. There are lots of things you can do, right? So finally, I just thank people working with us and also collaborators in the past maybe 10 years for all those work. Okay, so I'm stopping here. So thank you for you guys to listen that. If you have questions, I'd like to answer.
Thank you, Dr. Wang, for the presentation today. That was very interesting to see how your work in the lab can be help people in the future within the Alberta healthcare system. So from here on out, I will pass it off to the audience. If anybody has any questions, please, if you feel comfortable, unmute your mic and you can just speak, or if not, you can also type your question in the chat and I can read it out.
Yeah, it's a good. It's a really beautiful work, you know. It's really give us more in-depth cellular level, molecular level. So we are still particularly for coming school medicine. So we try to moving towards of the linkage from population database, EMR database, patient level to the really molecular level, omics database. So what your suggestions to moving toward that direction?
That's a really it's a really good question. I think this is one way we should moving forward. Like I mentioned, so UK Biobank already starting to sequencing those data, and they also have those patient level data. Basically, you need to build a model to combine these two different set of the information. And I think in the medical school here, as we are lucky have you guys, Hood, you're the leader for patient level data in the province, even in the national and international level. And we are also really good in the genomics part. I think we should work together in the future, more and more collaborations, and to push that forward.
Do you have the patient health number in your database?
Do you have what?
Patient health number. It's Alberta PHN.
We cannot access PHN, I think. Yeah, I think you guys can do, right? You can access.
Yes.
Yeah, so that's good. So one thing, for example, I can tell you another. We already get funded. So that is we will sequencing roughly 4,000 smokers, and half of them will be from Alberta Tomorrow projects. So means this Alberta people here. So we want to link those with other clinical information from the healthcare system, for example, to build a model to predict the lung cancer risk for these people. So that's the concrete example. So we can later on maybe we can access this participants' information from the Alberta health systems. But first, we will sequence them. We will sequence their blood sample.
So that's really good. We linked the approach Tomorrow database with administrative database already. So for defining hypertension, so that's really feasible.
Oh, that's really good. So we need to talk future to do some collaboration on this.
Yeah, if your genetic information analysis really prediction of the outcome of the patients, that really come from different levels. Yeah. Okay, thank you so much, Edwin.
Yeah, that's great. So next year we will put the sequence. Now we are collecting we retrieve the blood samples from the Tomorrow project for those people.
Hi, great talk. Thank you very much. So I was interested in your EHR autoencoder because that's actually something very similar to what we're doing.
Oh, that's great.
Yeah, because we're focusing more on the free text aspect of it. So we're using I don't know if you've heard of the BERT models for NLP.
Yeah.
Yeah, so essentially encoding documents and then encoding groups of documents to get something similar. You get a vector representation of it, just like you would for the autoencoder.
Yeah, yeah, that's another potential interesting collaboration for this. So our feature here is not from the test, but it's from the UK Biobank different items, so different features we used.
Okay, because it already is pretty cleaned up then, because I know one of the problems that we've always faced with extracting data from EHRs is that to find like comorbidities and things like this is not always the easiest thing.
Yeah.
Because maybe they're in a problem list somewhere, but those are undercoded. Or maybe if you have the right combination of tests, you can figure it out. Yeah.
That's really challenging using the test. I think they have some frameworks for the deep learning text mining something there, maybe help. And we also try to using that concept or modify that concept, put in the genomes. Because the genomes, now we think that's a test. No, that's a test, but this test is not that complicated than the real test we wrote. However, they have certain patterns in the genomes. They're pretty similar like tests. So we also try to model that to extract certain features in terms of like test. Let's say a word here, clinical in the test, right? But in genome, they have something pretty similar like this, but it's not defined yet. So here we define for each word, for each terms is already defined by us, human being defined. But in the genome, we believe they have such, we call it patterns or fingerprints there. So we need to find them out first and then define them and then put in the model in the predict model in the future. But I think text mining is still really challenging.
Yes, yeah, it's taking up a lot of our days. So are you using then the autoencoder representation as your features for, I guess, then predicting mortality, or that they'll get
okay. Here, so we predict the cancer risk. So based on the this is the features, we try to predict whether this person have the risk to get some cancer. So for this work is this. Of course, you can do many other stuff, but especially this one's for predict cancer risk.
And do you think once you have the genomic data on it too, are you also gonna auto-encode the whole thing together or separately to try to make a reduced reputation?
Yeah, this is an interesting question. So we will explore either the ensemble way, that's what you said, you do separately and merge them together to predict it, or infusion way. So they can be put them in different vectors, but could be in different weight with the EMR together and with different vectors, right? So all them treat with vectors together. That is infusion ways to do the predict model. But that is for our working objectives. We try to explore which one is better, or both are good. Yeah, two ways we will try.
Yeah, that makes sense.
Yeah.
And then do you want to try to use like GANs or like generative autoencoding to try to make or general adversarial networks to try to make sure you have great autoencoding representations?
Yeah,
I know they use that technique to try to make sure that you that your autoencoding representations are sort of robust.
Yeah. So we will try, different versions, autoencoding, and even certain other deep learning algorithms, even the CNN, for example, also. Because one of my students use CNN to treat different information, it still get a really good prediction. So autoencoder is really popular to treat those features here. But CNN is basically used in imaging, right? Imaging.
Mm-hmm.
So now we also try to do whether CNN can also to be applied here. This kind of data to do the prediction. That's also possible. And another one we are doing is for the deep graphs. So the graphs, the graph is a network. So so-called graphs is a molecular network. So in deep learning based networks, that's another direction. But all those things will be compared, modified, merged, and benchmarked to find the optimal solution for this.
Mm-hmm. That makes sense. Instead of CNNs, did you think of using more sequence models, like the ones they use for a language? Because like we think of the DNA as more of a sequence than like a two-dimensional representation like we have in images.
Yeah, that's what I'm talking about. This can be
Yeah.
can be modeled, like say the deep learning based, the text mining, whatever.
Mm-hmm.
That can be applied for sequencing.
I guess there's unlimited options really to choose from at the end of the day.
Right.
Thank you very much, though. Yeah, it was a great talk.
Thanks.
We have 5 more minutes to take any questions.
I mean, if no one else has anything to say, I have more questions. But
Go ahead,
I'm happy to let people
Elliot.
know. So at the very beginning, for your genetic regulatory network thing, so I just want to make sure I was understanding it correctly. So you have the network already mapped out, and then you essentially want to find like communities on that network, right? Because you do some kind of community detection, and then you see which communities are most sensitive to mutations causing cancer.
That's the traditional way to do.
Yeah.
First, like I said, people will generate, including us, will generate a lot of such single cell ATAC-seq data for the different cell types. Even these cell types, let's say for the same cell type in different development stages, their regulation can be different. So then we can build up so many different networks. Of course, we can model, let's say I want to model lung, right? Let's just, for example, lung diseases. So I can collecting all those cells in different stages, they come to lung, right? So now actually I tell you a real project now we're working on is for facial, the kids have different face, right? So we have even our lab generated they're from mice. So the single cell ATAC-seq data for different stages, they will develop face. Okay, so that's we can build up different networks, and then
so the mice have different faces.
No, so the network will be based on the face, based on the mice, but the face is from children, human.
Oh, okay.
Okay, so the face is from the human. So in the children's hospital here, I think in the world, we are the maybe only 4 or 5 places can generate 3D face from children or adults, doesn't matter. So, but now for children, because of the children's hospital, and different facial morphology and expressions, they will indicate more than, I think, 2,000 different diseases. But it's really hard to diagnose from clinicians. So the idea that can we using this network and link to this facial, these faces, this is 3D face, and then their genetic information to build up pretty model to the certain diseases. Okay. So you mentioned a certain, yes, a network community, network motif. This is in the past in the field, people have done. But now we want to push a new concept called digital genome version 2 here, that we have built up such networks based on different cells and whatever. And we will combine with sequencing information in a way of not traditional way thinking. We will in a way of DNA fingerprint in the genomes, and with even the network fingerprints. Okay, that's the concept not existing yet, but we think we will do that, push that direction. Totally new concept, new thinking for linked together. So that's not talking about just gene level. You say, oh, mutation there, detect a network community, they have many genes get mutated. This is, I show you in the past, right? We have done that before, but this digital genomes, we will forget about those genes. We will totally forget about genes. We will focus on the sequence, like somebody, you guys sequence a test, right? You forget about the terms, you forget the test, right? Same thing. So we will focus on those, and then combine with the networks, their regulatory sort of logics, because those have a relationship, have certain logics there. So the logics there plus this fingerprint concept instead of gene concept. And then we want to push a new model or a new concept in the field. So that's the idea for try to link to this.
So for example, you'll have your deep graph of your gene regulatory network, and then you want to be able to predict the face of the child. Is that it?
Yeah, the face, the difference. So let's say this. So let's say I give you one example. We are doing now is for they call the pellet. This is in mouse. You have certain defects there, right? So this is from the face. You can easily identify this kind of features there. So we want to see. We want to predict whether based on the information you can predict this thing linked together, right? So those things, because you can see easily, this is really easy diagnosis features. But for others, face development, other features, it's not easy to see from clinicians. So we can use those imaging from there, and also those to predict the disease, because their disease can be more. Many such disease is mental health related. It's a psychological, even psychiatry related. It's more neuron related stuff. Is you cannot see from the face, but you can see face. You can see minor defects there. But sometimes half of them the clinician cannot see. This is some diseases because of this, really fuzzy. So we want to use those together to predict.
So then in the future, we don't have to just watch out for facial recognition, knowing who we are, but also knowing our genes and underlying mental health status.
That's the purpose.
Cool. Thank you.
All right, so that's all the time we have for today's event. We're just about over 1 o'clock today. Thanks, Elliot, for the questions. It stimulated some really good conversation. I'd like to thank Dr. Wang and all of our audience members today for joining us on the last CHI speaker series of the year. We will continue in the new year, kicking things off with a second presentation from Dr. Russell Greiner. Keep an eye out on our Twitter and our website for future event announcements. And happy holidays, everyone, and we will see you in the new year. For more information on who we are and what we offer, visit our website at coming.ucalgary.ca/CHI. If you are interested in our services, collaborating with our team, or have any other inquiries, contact us via email at chi@ucalgary.ca.